In the last lab, we talked about meiosis and the creation of sex cells and sexual reproduction, and that the process of meiosis allows for us to not be identical to our siblings and parents. But there's certain patterns to genetic inheritance that we didn't really get into. We're going to get into those with this lab. But we're going to look at just the basics, or what we would call Mendelian genetics. Genetics in general is the study of all the inherited chromosomal materials, so all of the DNA that you get from your parents. And we can break these long chains of DNA up into segments or sections that code for proteins. We call these small coding pieces genes, and these are the things that are going to give us the characteristics we have. Our hair color, our eye color, how tall we are, what our voice might sound like, those can all be contributed based off of the genes we get. And the genes themselves are different in how they are shaped and sized and forms, and we call those alleles. Think of alleles like the different versions of that gene. For example, looking at eye color, there's genes involved for determining eye color, and some of those genes help determine blue eyes and some determine brown eyes. So all of us are a combination of different genes that gives us the unique look and appearance we have. Things like our eye color, whether or not we have freckles, and certain disease risks are all determined by the versions of genes, or the alleles I should say, that we have. But genes are not an end-all be-all. Many of the traits we have are inherited, but some of them are environmentally shaped. And for the most part, all the traits we have are factored in by both the environment and genetics. For example, skin color. Skin color is determined by genetics, the information that you got from your parents, as well as the environment. You have a baseline skin color that you have, but your skin can appear darker if you spend time outside in the sun. So it's a combination of two. Right? I'm a very pale person. Naturally, my genetics would determine that I can be very pale, but if I were to go outside and tan a bit, I would appear darker. When it comes to identical twins, they have the exact same DNA, yet they're not always going to be exactly alike. And this is because of that environment. Every single twin has their own personality, talents, likes, and dislikes, and even diseases can be different from one twin to another. One twin may end up with arthritis or diabetes or schizophrenia, and the other twin won't ever develop it. Although they have the identical DNA, there's external factors that will affect the outcome overall. So DNA is something we find in all living things. People, bacteria, trees, fish. It's the instructions for all living things. And like we mentioned in our DNA lab, that DNA is made up of nucleotides with 4 different bases: A's, T's, C's, and G's. But the larger units, the combinations of these nucleotides, we call genes. And these genes code for a protein or a piece of a protein product. The entire genome is all of the instructions, all of the DNA that makes you as a person, and it's the variations in these nucleotides, the different order that we can put them in, that gives us our unique nature and makes us different from every other living person on the planet. If we look at the human genome, again, we're looking at the entire genetic information collection in a person. It's about 3 billion base pairs long, and there's about 21,000 genes found within those base pairs. Now, not every single base pair is part of a gene, but every gene will be made up of base pairs. So not all of our DNA will code, so not all the DNA is part of a gene, but a good amount of it is. I know that I said that we're all very unique from each other, but it's a really small portion that's actually different. 99.99% of your genome is the same as everyone else that you know and everyone that you don't know. It's about the same with everyone else on the planet. But that 0.01% is significant enough to give us the variation we see in people. If you think about it, it makes sense. Everyone has, for the most part, has 2 eyes, and their face is structured the same, and the organs are organized in the same fashion, and you have 10 fingers, 10 toes, 2 legs, 2 arms, skin and muscle and bone. So if you think about all the similarities we share, all the way down to a molecular level, it makes sense that almost all of it would be the same from one person to the next. The real differences come in that 0.01% for determining things like hair color, freckles, disease risk, skin color, height, so on and so forth. So how do we get the genetics we have? How do we get the genetic information we have? We get it through reproduction. And depending on what kind of organism you are, that might be through a sexual process or an asexual process. Since we're humans, we use sexual reproduction to pass DNA on, meaning two individuals are going to come together to create a new offspring, and the offspring will be a combination of genetic material from both parents. This genetic variation is the result of both that recombination process as well as mutation, which we'll talk about in a little bit. The variation we have allows for individuals to have different versions of genes we call alleles that can create different combinations or different valuable forms of these genes. Like we mentioned, most things are influenced both by the genes and the environment, and for the most part, things are determined by many genes working together to give you the end product you see. For the sake of this class, we're going to keep it relatively simple, and we're going to focus on a single gene trait process. But just know for future classes that most things have multiple genes working together to create the end result. I mentioned mutation, but we didn't really talk about what it is. Mutation, if you're familiar with the term, may sound very negative. It sounds like it's something went wrong. But that's not quite what mutation means, although that can be harmful. It just means there's been some sort of sporadic or random modification of the genetic structure that can lead to new alleles. Some of these are induced mutations, meaning something happened to cause the mutation, like UV radiation, and others are just random. There's no rhyme or reason leading to that change. Now, we do have protective measures in place against mutations because many mutations can and are harmful. So our DNA has repair enzymes that can find these mutations by proofreading and repair any damage that occurs. However, not every single mistake is found, and so once the mistake gets through the replication process, it's locked in. Mutations, though, again, can be harmful, can do nothing, or can be beneficial. Ultimately, mutation is really the bare minimum we need to have variation within a population. It's the original source for all of the diversity we see in living things. So let's combine our understanding so far of inheritance and mutation. So we have a father and a mother here. Mother is healthy with no mutations. Father here does have a mutation noted in red. When they reproduce to create children, 2 sons in this case and 2 daughters, there is a 50-50 chance that a child will get either this healthy version or this disease version of the gene when it comes to the kids. Since mom is healthy over here and has no mutations, she has no risk of passing that mutation on since she doesn't have it. So every single child will get one healthy allele from their mother. But when it comes to dad here, there's a 50-50 shot that a child will get the healthy allele and a 50-50 shot that they will get the disease allele. So we see that half of the children then, 1 son, 1 daughter in this case, end up with the condition, and the other 2 are healthy with no mutations. Let's go a little deeper and get into some vocabulary terms. Now, the vocabulary for this particular section can be a little confusing, so you'll want to pay attention to the language being used and how it's being used, and who it's used for. The first 2 terms we need to be familiar with are genotype and phenotype. Genotype is talking about an individual's genetics. What are the genes that a person has? And when we're looking at a gene, we're looking at both copies that that person has. Remember that everyone will have 2 copies. Since we have diploid cells, everyone will have 2 copies of each gene. So we see them put into these combinations, these two-letter combinations, one of which came from the person's mom and the other which came from the person's dad. So these two letters are representing the copies of the gene that this person would have. So that is genotype. We're talking about what are the alleles found in a person for a certain gene. Phenotype is the physical appearance of an individual. So you can think of genotype. G stands for gene, P and the phenotype part stands for physical. That might help you remember it, but the phenotype is the physical appearance. This is what you look like. What is the outcome of the genes you have or of the alleles you have? So for example, you have brown hair or you have blue eyes would be your phenotype. And there's a few ways that we can pass information on. Not every gene is going to use the same pattern of inheritance when passing on information. We're going to focus mainly on the first one here, the dominant and recessive relationship, where one of the alleles is dominant over the other and it can mask the recessive allele. So in this case, we're talking about eye color, and we have a brown eye allele shown with a capital B and a blue eye allele shown with a lowercase b. This capital B can mask the lowercase b. So even though this person has a blue eye allele, they will have brown eyes since brown is dominant to blue. This big B here can pretend basically that this little b does not even exist. This is a simple dominant recessive pattern of inheritance. And for the most part, that's the relationship we'll talk about. But it's not the only way things can be handed down. There's also incomplete dominance, where heterozygotes, those who have the in-between genotype, where they have one capital and one lowercase letter, the heterozygotes phenotypes are blends of both alleles. So let's say we have an allele for straight hair and an allele for curly hair. If someone has both of these alleles, one straight, one curly allele, they have wavy hair, because wavy would be in between straight and curly. And then lastly, codominance, where both alleles are expressed equally. Our main example for this is blood typing when we look at the ABO blood system. Type A blood and type B blood alleles are both expressed as an AB blood type. There's no blending. We see both A and B alleles discretely. So what can we do with this information? How do we determine whether or not some offspring is going to have some certain trait? We can use what we call a Punnett square. A Punnett square is a tool that helps us measure the probability of inheriting particular traits. Basically, we're taking the genotypes of mom and dad, breaking them apart to their potential gametes, and then looking at the different gamete possible combinations here. Right? And this can be used by something like a genetic counselor who is a professional at using this genetic information to help people with determining what their offspring could look like. Not so important when you're trying to figure out eye color, but could be very important if you have certain diseases running in your family. So here's our Punnett square, and this is how we use it. We have dad here who is homozygous recessive, which means they have two recessive alleles. And we have mom here who is homozygous dominant, where she has two dominant alleles. So since dad here is homozygous recessive, he only has two little f's. Each sperm that he can make is going to be little f, and the other is little f. So they're both the same. For mom here, who's homozygous dominant, her eggs can be capital F or capital F. Again, no difference. And then we look at the combinations. We look at this egg with this sperm, this egg with that sperm, so on and so forth. And so we see this big F from mom gets carried over, and little f from dad gets carried over. This would be a potential offspring, capital F, lowercase f. We call that heterozygous because they have two different alleles. This individual here has the phenotype of freckles because freckles are dominant to no freckles. So big F is for freckles, little f is for no freckles. Since we have one dominant allele, freckles dominates over non-freckles. You'll be doing a handful of these Punnett squares, including some dihybrid crosses where you will be doing more than one trait at a time. Take your time on working on those. Make sure you're reading the instructions carefully, as there's a lot more boxes and it can get a lot more confusing. If you need help with those, make sure to attend the Zoom session and we can go into dihybrid crosses in more detail. We also want to get into how we can pass things down based on which chromosome a trait is found on. So we can classify genetic disorders and conditions based on which chromosome that gene is found on. Whether it's found on an autosomal chromosome, which would be chromosomes 1 through 22, or on one of the sex chromosomes, which we would call sex-linked, chromosome 23. When we look at autosomal conditions, they can be dominantly inherited or recessively inherited, and they affect males and females relatively equally. Sex-linked ones, though, typically are recessive, and they tend to affect one sex more often than the other. For the sake of this class, we're going to focus on the X-linked conditions, or X-linked inheritance, meaning we're looking at the X chromosome. When we're looking at an X-linked condition, we're looking at a condition that tends to affect males much more often than females. We can determine whether or not a trait is autosomal or sex-linked, as well as dominant versus recessive, by looking at what we call a pedigree. Here's an example of some sex-linked traits. So we have hemophilia, colorblindness, baldness, and Duchenne muscular dystrophy. These all tend to affect males much more often than females. But how do we figure this out? We use a pedigree. A pedigree is like a family tree that is describing the inheritance of certain conditions or traits. And there's some ways to read these pedigrees, and you'll really want to take some time practicing with these because these can be confusing. Punnett squares, for the most part, are relatively straightforward, but pedigrees can be confusing. So take the time to sit down with the pedigrees, and if you have questions on these, most definitely bring them to a review session. But let's look at one of the pedigrees here and talk about what we're looking at and how we can determine autosomal and X-linked, as well as dominant versus recessive. So here we have our pedigree, and we have a family tree looking kind of structure. So circles will always be females, squares will always be males. So this would be grandma and grandpa. And we see in all the individuals in a line are going to be from one generation. So here's one generation here, and then our second generation, and our third generation. We call this line linking this circle and square a marriage line because it denotes that these two are in a relationship and have children together. The children branch down from that marriage line, and we see that this couple here has 2 daughters and 2 sons. If this circle or square is shaded in, that means this person is affected. They have the condition we're studying. If it is white, then they do not have the condition we're looking at. For the sake of this class, I'll keep it simple enough of affected versus unaffected, but in your studies, you may come across some pedigrees that show the symptomless individuals. These are individuals who are unaffected by the condition but carry it, meaning they do have an allele for it that they can pass on to their children, even though they themselves don't have the disease. So here we have grandma who does have the condition, grandpa who does not. All of their children are symptomless. They're all carriers. This woman here has a marriage line to this man here, so she is in a relationship with him, and they have 4 children themselves, one of which is affected, 2 are carriers, and 1 who is completely unaffected. When you're looking at pedigrees, you want to look for the following information. You want to look at the ratio between males and females affected. If it skews heavily towards males, meaning many of the males are affected while very few females are, it's probably X-linked. But if it's a relatively constant number, meaning like about an equal number of males and females are affected, then it's probably autosomal. It doesn't need to be perfect. It doesn't need to be like 4 and 4. But if it's 4 and 3 or 5 and 6 that are affected, that's probably autosomal. We also want to look at the skipped generation. The skipped generation is when we follow from these parents here, we're looking to see whether or not the children are affected. If something is recessive, then we can see a skipped generation, meaning one of the parents has the condition, the other parent does not, and that the children they have, none of the children necessarily have to be affected, and that the children have, the grandchildren, you can see that trait pop up again. So if you were, let's say we trace here, this would be an example of a skipped generation where grandma has it, but this daughter here does not, and yet the granddaughter does. So if we trace the bloodline from this granddaughter up, neither parent has it. Trace grandmother up, grandmother does. Between the 3 generations, we have one skipped generation. That tells us that this is recessive. If it was dominant, this individual would have to have a parent with the condition. The reason being is that if one of these conditions is dominant, having one allele that gives the condition is all you need to be sick. So the heterozygotes, the ones that have 2 different alleles, will always have the condition if the condition is a dominant inherited condition. I'll put out a worksheet for you guys that you can practice with that has some answers. That way you get a little bit of practice and a little bit of insight into what these pedigrees are and how they are used and what they look like. Again, you want to get as much practice as you can with both the Punnett squares as well as the pedigrees, because these both, especially the pedigrees, can be very confusing, and you will see at least one of these on one of those exams.